So finishing up that last part, each motor neuron innervates its muscle fiber at the neuromuscular junction, and the neuromuscular junction is the axon terminals of a motor neuron plus the motor end plate, the modified sarcolemma, plasma membrane of a muscle fiber. So the combination of those two things is the neuromuscular junction. On the slide, histologically, you see the individual muscle fibers here. The black lines are the motor neuron axons, and then they branch and reach the neuromuscular junction. So these little dark spots that you see here are the axon terminals in blue here. And just below them is the motor end plate of the muscle fiber. So those two things, axon terminals of a motor neuron and the motor end plate of a muscle fiber, comprise the neuromuscular junction. So the synapse is the junction or the meeting of these two structures, axon terminal and the motor end plate. And the gap between the two, there's a thin little gap, is called the synaptic cleft. The axon terminal or synaptic knob or terminal boutons, another name for it, contains synaptic vesicles with acetylcholine within them. The motor end plate, the modified sarcolemma, is highly convoluted and has receptors for acetylcholine. When the action potential reaches the axon terminals, the vesicles release the acetylcholine diffuses across the synaptic cleft and contacts the acetylcholine receptors on the motor end plate. And then we'll talk about what happens next. There's an enzyme here in yellow called acetylcholinesterase, and it's an enzyme that breaks down the acetylcholine as it's diffusing across. So the function of acetylcholinesterase is to make the effect of the acetylcholine when it's released short-lived, transitory. And there's also pumps that will pump acetylcholine back into the axon terminals. So in other words, for the result of these things is that a muscle fiber will only contract if there's a steady stream of acetylcholine being released from the axon terminals, meaning a conscious steady stream of action potentials, conscious wanting to contract the muscle, is constantly reaching the axon terminals. So in summary, the nerve impulse, which is initiated in the brain, goes down the spinal cord, spinal nerves, motor neurons, action potential hits the axon terminal, acetylcholine is released. Acetylcholine diffuses across the synaptic cleft. Where's my cursor? Here we go. Attaches to receptors, depolarizes the motor end plate, causing an action potential there. The action potential shoots across the sarcolemma and then goes down the T-tubules, diving into the muscle fiber, just like Luke shooting down that canal down the Death Star. Episode 4, probably still the best one. Okay, so the action potential down in the T-tubules causes calcium to be released from the sarcoplasmic reticulum. The calcium binds to troponin on the actin thin filaments. The tropomyosin shifts, exposing myosin binding sites. That allows the myosin head from the myosin thick filaments to latch on. That's cross bridge formation, or cross bridges form. The myosin head then pivots, and that's the power stroke, causing the thin filament to slide past the thick filament in opposite directions on either side of the M line, either side of where the thick filaments are located. The thin filaments will slide past the thick filaments as the myosin heads ratchet and ratchet and ratchet, like that little video clip showed you. These Z lines will move closer to each other as the thin filaments are slid past the thick filaments. That means the sarcomere contracts. That means the myofibrils contract. That means the muscle fiber and the fascicles in the entire muscle will contract or shorten. And then actin binding to the myosin causes release and re-energization. Wow. Energization. Energizes. I mangled that, didn't I? Energizes the release and the cocking of the myosin heads, so that myosin head can then form a new cross bridge. Okay, so then sarcomere shortens and ultimately the entire muscle shortens. And this will link here to that third video I showed you a little while ago. So as you see here, as the amount of overlap increases, thin relative to thick filaments, the Z lines move closer to each other, the sarcomere shortens. Down here, we see at the bottom, this is relaxed, extended muscle fiber, or myofibril in this case, within the muscle fiber. The amount of contraction is increasing as we go up. The way you know that is the lighter I band is getting shorter because the thin filaments are overlapping more with the thick filaments. Notice that the darker A band stays the same length because that corresponds to the thick filaments. The H band in the middle of the A band starts to disappear as the amount of overlap increases and increases. Overlap of thin versus thick filaments. Okay. And then this little dark line in the middle of the lighter I band is the Z line or Z disc. And notice that it gets closer to each other. They get closer to each other as the sarcomere contracts. And again, all of the sarcomeres are linked together within a myofibril, causing the entire myofibril and then the entire muscle fiber and the fascicles in the entire muscle to shorten. Wow, relaxation. So muscle contraction ends when the action potentials are no longer generated. In the brain, they're no longer generated. You no longer want to contract a particular skeletal muscle. There's no action potentials leaving the motor neurons out of the spinal cord. There's no action potentials hitting the axon terminals. There's no release of acetylcholine. Any acetylcholine that would be in the synaptic cleft diffuses away, gets pumped back into the axon terminals, and or is deactivated, degraded by acetylcholinesterase. Because of that, calcium is no longer released. It gets reabsorbed back into the sarcoplasmic reticulum. Calcium is no longer able to bind to troponin. Tropomyosin closes the myosin binding sites. Cross-bridge formation then is no longer possible. The muscle relaxes, going back to the extended length. Okay, motor units. A motor unit is one motor neuron and all the muscle fibers it innervates and controls. So here we see 3 motor neurons and the color-coded here for you, the muscle fibers that they control or innervate. That means this is showing 3 motor units. Over here, 1 motor unit. There's its one single motor neuron and the muscle fibers it controls or innervates. So a nerve impulse will cause contraction of all the muscle fibers to contract in unison, what's referred to as the all or none principle. If there's an action potential coming down the motor neuron, its axon terminals will release acetylcholine, causing the whole sequence of events for contraction of the fibers. Okay? That doesn't mean that the entire muscle contracts. Only the muscle fibers controlled by the motor units that are activated. So if you think of it, let's say you go to the gym, you're going to lift some weights to do arm curls, and you pick up a 5-pound dumbbell, 5-pound weight, and you flex your elbow and you curl it, right? You lift it up and down. 5 pounds is not very much, so you don't activate all of the muscle fibers in your biceps brachii because you don't need to. It's not that heavy. Only relatively few motor units are activated or recruited because you don't have to lift that much. Okay, put the 5-pound weight down, pick up a 10-pound weight. Okay, now it's more work. You have to use more of your muscle. You have to apply more force. Consciously, more motor units are being recruited, more muscle fibers because it's heavier, more work. Put the 10-pound weight down, pick up maybe a 30-pound weight. Now it's heavy. Maybe you can lift it 3 or 4 times and that's it. You're pretty much at your maximum. You're using the entire muscle now. Full force, as much as you can contract, as strongly as you can contract it. That means you're activating all the motor units in that muscle. All of the motor neurons that reach the entire muscle have been activated, fully recruited. You're lifting as much as you possibly can. Okay? There's also an inverse relationship between the size of the motor units and the degree of control of a muscle. So, precisely, finely controlled muscle movements like the movements of your eyes or say your fingers, your digits, is possible because there's relatively small motor units involved that are innervating, controlling relatively few muscle fibers. So they're not really strong muscles, but they have a lot of fine control. The opposite of that would be strong, powerful movements controlled by strong, powerful muscles like the muscles in your leg and thigh. Right? Think of like running up the steps. These are large motor units. In other words, each motor neuron can innervate up to hundreds of thousands of muscle fibers. So these large motor units give you a lot of power, but you sacrifice fine muscle control. And then recruitment is what I was talking about. How many motor units you're activating to lift a particular item or to generate power from your muscles. Okay, a little on muscle tone. So even when your muscles are at rest, in quotation marks, some of the skeletal muscles are contracting and relaxing and contracting and relaxing, even though we don't really actively think about it. This provides what's called muscle tone and gives us a posture. If you're sitting there, you're activating muscles in your neck and in your back and your torso, etc., to maintain posture. When you're standing upright, the muscles in your legs, your back, your neck, etc., help you maintain posture, and your muscles are constantly contracting and relaxing, contracting and relaxing. So this is what muscle tone refers to. And it also keeps your muscles healthy in a physiological sense so that they can work well, especially when you really need them to consciously. Okay, muscle size, muscle hypertrophy. You can make your muscles grow larger and stronger, and it's not by making more muscle cells, but by making more myofibrils within each muscle fiber, increasing the diameter of the muscle fibers and thus increasing the diameter and strength of a muscle by working them vigorously. What happens is the larger muscle, which is now more powerful, can do more work, needs more mitochondria, more myoglobin, a oxygen storing protein, more glycogen storage is possible, and the blood vessels are increased. You have more blood capillaries, so you can provide more oxygen. The mitochondria can utilize glycogen to break it down into glucose to make plenty of ATP to work those more powerful muscles, and then myoglobin providing more oxygen storage in addition to the oxygen provided by more blood capillaries. Muscle atrophy is the opposite. Muscles shrink in size and get weaker. If you break an arm and you have a cast on it for 6 to 8 weeks, you take the cast off, that arm that's healed is not as strong as the other one, If the other one's been used and that arm that's been in a cast has not been used, the muscles are smaller, they have atrophied.